Today we are going to see some statistical aspect of bioequivalence study. In this basic structure is to understand what is the equivalence, and in a statistical aspect, if we have to test a equivalence hypothesis, what could be the possible statistical solution for that? We will see a computational aspect also, but I would like to make the video in two parts. First, we are going to see that what are the statistical techniques and how the decision making process involved in equivalence testing. And then later in part two, I would like to demonstrate a computation process using SAS system. So let's start it. So basically this is needed in a generic drug discovery, but in principle, wherever you have a requirement to test a equivalence with some kind of interval, you can use that. So in a generic drug, first let's understand how this mechanism is working. So generic drug is simply a copy of a research drug, which we called a innovative drug. So innovative drug has a pathway of research from molecule to the drug. There is a several phases, several regulations, and so many things on safety aspect and on efficacy aspect has been tested and approved by a regulatory agency, which is country specific where you want to get approval. Now, it has a patent life so that the company who has developed, they can get recovery for their investment. But after the patent, anyone is free to develop this drug by their own and start selling it. So they don't have to go through the rigorous approval process and rigorous research process. They just have to see that what I have developed is basically equivalent to the innovative drug. Now, how this works? Let's see. Two concepts basically. First, when you take a drug, what happens? So basically, there is two component. One is when you take a drug, what drug is doing to you, and once drug is inside your system, what body is doing to your drug. Okay, so when we take the drug, basically what body do to the drug is called ADME. Means it's absorb the drug, it distribute inside the circulation system, it metabolize the drug, and finally it will filter it out and excrete the drug. That's called the pharmacokinetics profile of the drug. But once drug is going through this ADME process, it do something to your body. Like it will reduce your fever in case of you are taking a fever drug. It may shrink the tumor, it may heal your disease, or it may create some adverse event reactions like that. So that is called pharmacodynamics effect of a pharmaceutical ingredient. So these two are PK and PD. What is mostly we are interested in PD. When we take the drug, we want our disease to be healed. That's basically PD. And testing a PD is a very rigorous tough process where any research company do from a molecule to the drug, developing the drug. But scientifically, it is believed that if any drug has same PK, any drug, when I say that, it means the same molecule or same component developed in different profile, different formulation, different ways, but having the same mm, chemical ingredient, I can say. So if they have a same PK means absorption, distribution, and metabolism, then their PD effect, like healing effect, will be the same. That's the scientific belief and was well proven in the science. So we are taking help of this fundamental principle, and then what we are doing is basically we are just proving that my newly developed formulation is having the same PK with the innovative. And if I am equivalent to that, I can say that PD effect will be same, and I can again start getting approval and marketing of my drug. That is called generic drug. So we must prove bioequivalence for generic with original innovative drug before getting its license. Now, what bioequivalence and how it's work? So bioequivalence refer to the similarity in the bioavailability of two drugs. Now, what is bioavailability? So it's refer to the rate and extent which active ingredient of drug is absorbed and become available in the systemic circulation. How we measure it? So the rate and extent of a drug absorption and distribution, like at what time, how much drug available in blood, and in totality, it demonstrates by providing area under curve. So suppose you take the drug and after 5 minutes you measure how much drug available in the blood, then 10 minutes, then 20 minutes, then 2 hours, 4 hours, and so on up to a day or two you measure. So you will sum it up all the area of different time and you get area under curve for the whole drug availability in your blood. And then there may be a point when you observe that yes, at this time we observe maximum drug concentration in the body or the blood wherever you're measuring. So that's called Cmax of the drug. And the time when Cmax reach is denoted by Tmax. So to prove a bioequivalence statistically, we have to prove an equivalence on bioavailability on the above mentioned measuring parameter, mostly AUC T. T means up to time T, what you measure the concentration. And then there is a AUC 0 to infinity. So it's impossible to measure up to the time infinity practically. So what we do, we actually measure the elimination phase and we try to say that what is the slope, and statistically we will extend it up to the time infinity when actually slope go and touch the time axis at zero concentration. So that area is denoted by AUC infinity, then Cmax, and but sometime also Tmax we want. So mostly it's a AUC and Cmax, but couple of time Tmax. When Tmax, there is another principle why. Now just let's see from this uh, demonstrated visuals, like see I'm using a pointer here. So this is the time when drug is administered and the concentration is absolute zero at this time. Now the time progress here, and at certain frequency you are measuring the concentration. So this is absorption phase. Drug is getting absorbed and it is becoming available and it goes on with respect to the time. And here you reach the Cmax, maximum concentration. At this time, this is the Tmax you reach. And then elimination phase is started. Basically, this period also the distribution, absorption, elimination, both are happening. But absorption is very high compared to the elimination. And then drug got absorbed, max. But after this Cmax point, the distribution, means elimination is at the same rate, but there is nothing more to absorb. And then you see the curve declining and it goes on eliminating the drug through the kidney filtration or any other filtration which wherever the drug is targeted. And then sometime this is the max time we measure, but after that we leave it. And then theoretically if we can extend this slope somewhere, it is going to touch the line at zero, and this is the AUC infinity we call. So you see this whole area is AUC T, and if this whole area extended till infinity, AUC infinity, this is Cmax, this is Tmax. And these are the parameter is going to demonstrate how your drug is basically distributed in your body, at what rate, how much exposure, at what time maximum exposure. So rate and extent of the drug inside the body, the whole distribution is going to be taken care in this three profile: AUC, Cmax, and Tmax. Now there are a couple of things as a PD, like to observe a effect, this is the minimum concentration need to be body. Then only drug will show some effect. So that is one PD, what drug will do. And then this is the maximum safe concentration, means up to that the drug is safe, but if concentration reach beyond that, then it become toxic, and that's why it is uh, maximum safe concentration. So MSC, MEC, it's called of abbreviated name, but you should understand the concept here. And then many other PD parameter based on our disease of interest. But in PK, we are going to use these three parameters to prove that we are equivalence. So you understand how we are measuring the bioavailability. And then we are going to prove that we are equivalent in terms of bioavailability, and that's why we are bioequivalence. And then we expect the same effect on PD end, and hence we need permission for marketing and approval. Now, see, if I am giving a drug which is patent, like reference, I can say, with whom I'm going to compare, I have developed, say, this one as a test. So this is plotted, the whole concentration. So you see that suppose I am getting n number of subject and I'm measuring their concentration at 0, then half an hour, 1 hour, 2 hour, 3 hour, and so on. So taking the average mean of concentration time wise for all the subjects and plotting for test and reference. So you can see the profile, and this is like 0 to 1200 at 12-hour time. This is the profile for test and reference. This is called linear scale. And when see at this point we don't understand very much well how the drug is working. And basically in this point, we need a lot of sample between 0 to 2 hour. So what we do, we generally expand the axis in a logarithmic view. So it's called a semi-logarithmic axis, and we have extended it like a fold of 10. So this is 0.1, 1, 10, 100, 1000, like that. And then we see the profile. So now you can see that the initial samples are very clearly visible where it's going and how it's going. So that's the way of visual demonstration, and to understand how the concentration flow is going. But overall, understanding the whole profile, we have to consider all three parameters. So let's understand, once we are going to prove the bioequivalence, what are the criteria to basically claim that we are equivalent. So as a thumb rule, you are not going to find any major reference for this, but couple of places it's mentioned that we consider a 20% range, roughly 20 or some plus minus 20. If we are in 20% range of the original drug in terms of PK parameters, we are basically equivalence. Now, the big problem with the PK parameters are their distribution. So if you can see here, this is the concentration, and we have taken sample with respect to time. It's not looks like normal. Here also you can see the profile not looks like normal. The same thing happening once you will get aggregated area under curve, Cmax, things like that is not normal. So our statistical techniques directly not applicable here. Most of the statistical technique which assume a normal distribution theta. So there are a lot of background principles and theoretical development are there to understand how to deal with this data. But majority of the scientists agree that the PK parameters are log normally distributed. So once you will take a log, it will become the normal, and then you can apply all the principle. So how this 20% plus minus will work on a log normal scale? So then we have figured it out a threshold on a symmetrical level. So two things. Once you want to know that what is the difference between two drugs. So on a linear scale, it's a difference, a minus b equal to 0, and then you say a and b is equal. But on a log scale, it's the ratio. So a by b equal to 1, basically. And if you take the log, it means log a minus log b equal to log 1. That is going to be 0. So in that way, they have figured it out how symmetry can work, and there is a 2 point, 0.8, 1, and 1.25. And we take the exponential of that. So this point has been derived on the log scale. And once we get them on the original scale by getting the exponential of it, so we are going to found exactly the symmetrical axis around 0. So minus 22 and plus 22 point some floating numbers. So we can say that if we'll take a boundary of 0.8 to 1.25, somewhere we are testing it on a plus minus 22.31% range. And uh, that's exactly the boundary has been set for claiming the bioequivalence. So if we see the FDA guidelines, EMA guidelines, and Indian health regulation guidelines, all the agencies are fine if we are in between these boundaries, zero point 0.8 to 1.25, when the data is analyzed on the log scale. So let's see how we are going to work with this scale. So we have designed a study, and we have taken a couple of subjects, and we made them in a two sequence because we are going to randomize them in a two different way of administrating the drugs. So sequence A is drug A and B, and sequence 2 is drug B and A, like in a crossover. So if you say that test and reference, so sequence A is going to get test, and then there will be washout and then reference. Sequence 2 is going to get first the reference, then washout, then there is a test. So we make crossover, like two period, two treatment crossover design. So what it will say that on the same subject, test drug has been given, then the washout period has been given. So whole concentration of the test drug has been washed out, then reference has been given, and we have evaluated the profile. So it means like maximum effect of occurring a bias has been taken care because it is the same subject. Only the effect which drug can have a different concentration of different output is due to the subject itself. Otherwise, rest all thing has been taken care. So that's why a crossover design is having more controlled on the bias occurring from outside. And the similar way, suppose if the sequence may be doing something like giving test first and then reference, or giving reference first and text, we have just half of the subject has been getting the same thing in a reverse way, reference and test. There is a period one, when you get test and reference both administered in two different arms, and then in period two, it's swapped. So you can say that out of your subject, 50% will be in sequence A, who will receive test and reference both, 50% in sequence 2, who will also receive test and reference both, but in a different period. So every subject here has test and reference administered. And that's the design part. We have controlled maximum biased and maximum external source which can have effect on the outcome is controlled in this design. Now let's proceed to understand, what hypothesis we want to test. So the hypothesis here is the drug concentration data obtained from the above proposed study design, which will facilitate the calculation of PK parameter, AUCT, infinity, Cmax and Tmax for each subject for each treatment he received. So every subject will have two PK parameters. So let's say subject 1 get test and reference. So now for the test, for the subject 1, I have calculated his AUC T, then AUC infinity, then C max and T max. So subject 1, what he got in first period is test, and for that this AUC T infinity C max T max has been calculated. Similarly, what he get as period 2 as a reference, this PK parameter has been calculated. And this is so on for every subject. So every subject has two pair of data. One is for test, one is for reference for each PK parameter. And that's what we are going to test, that whether these two pair of data is similar or not in terms of bioequivalence interval. So on a log scale, we have fixed the boundary 0.8 to 1.05. So my hypothesis here is T minus R is greater than my boundary. So either in plus or minus, but it is greater, versus alternate is it is in between minus theta to plus theta. But once I converted in the log scale and then I want to test, so suppose my average PK parameter on test, so T by R ratio is less than psi 1, or it should be greater than psi 2. That's my null hypothesis, versus alternate is it is in between. Okay, so this value of minus theta equal to this on log scale, I can say you can see this anomaly and similarity of that. Now this boundary we are going to use consistently for every calculation. So if you can see here, the basically we are testing a composite hypothesis. My hypothesis is either I am less than this or I am greater than this. This is my null hypothesis. And if both are rejected, means I'm this less than psi, T by R ratio less than psi is rejected. Simultaneously, T by R greater than psi 2 is rejected. If both is rejected, then I can conclude that yes, T by R ratio may be in between psi 1 and psi 2. So null hypothesis is a composite of two, alternate is one. That is my conclusion. To test this, there is a test proposed. So Suriman's test, two-sided test proposed. That is a t-test, but again, it's one-sided. Now, if we are going to see state, there is a concept of at what level of alpha. So the concept is that even we are testing two hypotheses, one is less than or greater than, but both cannot happen at same time. Suppose under H naught, I will get a value of T by R, but that will be either less than psi or it will be greater than psi 2, but it will both simultaneously will not exist. That's the tricky point. So I am testing here the two hypotheses, but my error will be only coming from one, either from this or from that. Okay, so the error is either from this or from that. So even testing the two hypotheses, my error is coming from only one. So if I will get a 90% confidence interval also, or if I will test at a 10%, the actual error commitment will be 5%. And that's why when we take the boundary, we take the 90% of confidence boundary rather than 95%. And even with the 90% confidence boundary, our overall error commitment rate will be within the 5%. That's the trick point here. So let's go ahead with the statistics. Statistics is proposed the toast for equivalence testing. This is two one-sided test, and the toast procedure and upper and lower. So say L and U are going to test. Equivalence boundary is specified. and then you have specified your composite hypothesis and want to test it for rejection. So once you reject both, you concluded your parameter theta is within the L and U, and that's exactly going to conclude your equivalence. Now see how we are going to do it. So we are going to do it not only with the directly applying the t-test proposed by the Sorimans for two one-sided in such a situation, because come to the design, see that if there may be slightly possibility that period may have something effect, or let's say that washout which you have taken is not fair enough, so still some amount of concentration is remain in the subject body or anything, or maybe subject itself is responding totally different at time period 1, and he is responding totally different at time period 2. So getting a pure hypothesis test from t-test to one-sided toast will not give you a very good idea. So we'll go with the model-based approach. Now, how we are constructing a model is like log of pk parameter, that is yijk, equal to first the intercept. This is the part of intercept, but you see a I here, I will explain later. Then alpha into the treatment effect, beta into the period effect, and gamma into the sequence effect, plus epsilon. Now, subject is say I equal to 1, 2, 3 up to n, and every subject may have a different intercept, and that's why subject as a random component we will take, that he may randomly respond something. Every subject may be randomly have some different response, so overall intercept has been adjusted by the random component of the subject effect. Okay, treatment in our example it's only 1 and 2, j is equal to 1 and 2, period also 1 and 2, but basically for high variable drugs there may be multiple period, and a sequence here also in our example we have 1 and 2, but there may be multiple sequence. Like working with a very high variable drug, you may have a design like 4 period 4 sequence full replicate crossover design. So your sequence may be AB, AB, AA, BB, or BB, AA, BA, BB, something like that. Now, if you understand the design, so our basic principle here is that from this model, we will get the contrast and we will estimate the least square mean uh, for the treatment. Okay, so if we will estimate the least square mean for the treatment, so basically that mean is adjusted by the period effect and the sequence effect, and that will be really adjusted mean. We will get the mean, and then we will basically get the difference between the least square mean of treatment 1 and 2, or treatment test versus reference. And this difference is on log scale like log uh, a minus b, but in actual scale it's going to be ratio, t by r ratio. So we will get the estimate done from the model, and then we will back transform it to the original scale, and we will present it as a t by r ratio along with their confidence interval. So in this whole example, we are not going to work with the p-value. That's the goodness. If you will estimate p-value also, definitely it may come significant, just like when you work with the odds ratio and you try to see that whether your confidence limit is crossing the 1 or not. If it is crossing, it means you are not going to get a significant result, but if it is not crossing and whole interval is only on the one side, then you are going to have a significant effect. The similar concept apply here, that if your T by R ratio is uh, containing 1, but it's the whole confidence interval is in a one side of your threshold, then you can say that rest is assured. So we are going to work with the confidence interval. So now how we are going to present the result? This is uh, I can say mock cell, like you have to work with your data, you have to get the result estimated out, and then you have to present in such a style. So you see, I have presented my PK parameter AUC infinity, AUC last, Cmax and Tmax. Then my treatment TR for every parameter. How many subjects are there? Like once you will work with the concentration data, what will happen? You started with the say n number of subject, but couple of them didn't provided you the blood or something happened. You cannot get the PK parameter estimated for that subject. So this is the n star, which will give you basically that how much subject has this parameter available or basically calculated. Now what you are going to do here is you are going to estimate the geometric mean. Why geometric mean? Because you have taken the raw mean, like adjusted mean, least square mean from the model on a log scale data, and you have converted it back to the actual scale. So it's become basically the product divided by their nth root, and it's become a geometric mean. So we take the geometric mean, it's a standard error. Maybe you can present the confidence interval also for each least square mean, but both are works fine. So that is estimated. And then we are going to work with the T by R ratio. So this by this, we have estimated it, geometric mean ratio, we call it, and the lower bound and upper bound. And what we have to test here is that it should be within the range of 0.8 to 1.25. Uh, in the footnote, we have provided that how my model has been constructed, and it is log transform having subject as a random component, period and sequence as a fixed effect, treatment as a fixed effect, and that's what my model has been constructed. And then for T max, there is a tricky thing because you see here T minus R. So basically the T max data is a time data, and it do not go with the log normal distribution. Basically it's not even normal or things like that. So we have to apply a non-parametric test for Tmax, and that's why here you see that uh, for Tmax median is presenting under adjusted geometric mean. Hodges-Lehmann estimate has been calculated and its confidence interval has been presented. So this is a copy of one of the uh, corporate who work on this equivalence trial, and they would like to present their results to the authority in such a way format. So just for demonstration, I'm saying that let's see, we have observed a profile like this. So maybe green one, you can see that maybe our reference, and the blue one is our test. And we have presented their profile along with their standard error or standard deviation, whatever the best you can see for a visual display. And then you see that how their profile is going on. So this is in total profile with their standard deviation, variation, and you can easily see that whether it is going to be equivalence or not. So it's a visual comparison to understand the equivalency. Now, how it is computed? So in a SAS program, it is computed with the below mentioned code. So I'm using a mix procedure, that is a mix model. My data is plugged in here, and then method I am giving a type 3. There is several method to estimate uh, contrast and LS mean, and compute the uh, p values and compute the confidence interval and estimate the difference. So in that there is a restricted maximum likelihood method, there is a type 1, type 2, type 3. So these are going to work basically with the ANOVA and ENCOVA setup. So when I use type 3, it's here as an ENCOVA setup, taking care for random effect of the subject, and then by PK parameter. So for every PK parameter it's going to work. I'm not going to pass my Tmax data here. It's only AUC T, AUC infinity, and Cmax. And then the class variable is basically saying that the value in these variable, don't treat it numerical. Even though there will be one and two value, but these are the class variable, nominal variable. So you don't compute any statistics for that in terms of treating it continuous, but basically it will treat as a class variable. My model is my pk value, which is log transform, is dependent on treatment, period, and sequence. And then I have plugged in, like I'm going to calculate the confidence interval, residual covariance matrix, correlation coefficients, and I want a solution. Solution means basically the estimates of the beta coefficient for each of the dependent variable, each of the independent variable, which is passed in the model. And my degree of freedom has been calculated from Kenwan-Roger approximation. That's what this code is saying. And I am going to get all the data out. Uh, so for that, couple of another syntax I have to give, like outpred is equal to the residual and predicted value has been given in this data set. Subject as a random component. This special text is to make the computation based on consistent with our design. So our design is 2 cross 2 within the treatment. Like every subject has two treatment, test versus reference. So that's what is saying, that subject nested for the treatment. It means that each subject has each treatment which is nested. And then I'm going to estimate the least square mean for treatment. pdiff is basically going to calculate the difference between the least square mean, their p-value, and if you want to control, like you have multiple treatment and you want to see that I want to compare every treatment with reference, so you put a control, then comparison will be control versus every treatment. Otherwise it will do comparison of all permutation and combination of the treatment, and this is for confidence interval. I want to estimate my test versus reference. So I'm passing the design matrix for my treatment, basically two treatment, and this is 1 minus 2. So it is going to take the difference between first and second. It's alpha, and here see alpha is 0.1. So at that level of confidence, I'm going to predict the estimates. And these are ODS output, means ODS in a data, my estimates in this data set, and LS mean in this data set, covariance parameter in this data set, going to run the model. That's the SAS code for that. For SAS code for non-parametric test for Tmax, Haus's-Lehmann estimate. This is the data. Non-parametric test procedure, plug in the data, Haus's-Lehmann estimate. Class again treatment, variable is a pk value. Here pk value only the Tmax. Now I'm going to compute the exact confidence interval Haus's-Lehmann and ODS and select Wilcoxon score, Haus's-Lehmann score. This text is for the graph, which you see that profile of the treatment concentration versus the time curve. So on a XYZ, little bit work with the cosmetics and a layout of the graph. So basically it's a plot. I'm plotting a scattered plot, time versus the concentration. Here concentration is mean concentration you have calculated, and by group. Group means treatment. And then my error bars is coming from this code. And then this is again a series. So series means it's going to connect your dots, nothing else. Then this is the title, and x-axis what label, what should be the value, minor axis, major axis, tick mark, that's all. And that's why you will generate the graph. Now generated a data, that is a hypothetical simulated data, and try to compute this code, and this is the output I got. So you see my AUC is somewhere reference test this, standard error, and then T by R ratio is 0.97. My lower confidence interval is 0.87, upper is 1.08. So you see this lower and upper both are within that boundary. So I can say, okay, for AUC T, my drug is equivalent, but what happened for Cmax? So you can see here, Cmax, the data has been given, but Their T by R ratio 1.13. Okay, still I am in the boundary, but when I take the confidence interval, upper limit is crossing the upper threshold. So it means that it is not equivalent, it's going beyond. And for a confirmatory bioequivalence, we should be equivalent on all the PK parameter. That's why this simulated study is failed. And this is we conclude. So now you have fair understanding about theoretical aspect of doing a bioequivalence study. And that's all from my end for part one. Let's meet you at part two.